Okay, so this mini lecture is going to be on 5.3 and 5.4. We're going to talk more about the dermis and the pigments that contribute to skin color. So we already touched briefly on the dermis and that it's the second general layer of the skin and it lays deep to the epidermis. It's predominantly made up of connective tissue and has a lot of properties of connective tissue proper. So it's going to include the same cells that we're talking about if we're talking about CTP. And that's going to be fibroblasts, macrophages, and sometimes we'll see mast cells or leukocytes. And because we're talking about connective tissue, we're talking about cells in a matrix. So that's what we're going to have here. And in addition to that, remember that the dermis is vascularized and innervated. So we're going to see nerves and we're going to see different blood vessels as well as lymphatic vessels, which we'll learn about in A&P2. We also are going to have our hair follicles housed here, sebaceous glands, and sweat glands, all of which you identified in lab. And the dermis has two specific layers, your papillary layer and your reticular layer. So the papillary layer, as I mentioned previously, is called the papillary layer because it includes what are called dermal papillae. So these finger-like projections of the dermis into the epidermis. And these finger-like projections usually contain some sort of sensory component. So we learned about Meissner tactile corpuscles in lab, and you would find them right there. We also have some free nerve endings, as well as capillary loops, so some sort of blood supply in those areas. I do want to point out that the Meissner tactile corpuscles that you identified in lab on the integumentary skin model, they are distinct from the tactile or Meissner cells that are found in the epidermis. So one are receptors, so those are the ones that are in the dermal papillae, and one are actual cells, not a receptor structure itself. This area is made up of loose connective tissue, specifically areolar, which is really great because just in the event that we penetrate our epidermis and go down to our dermis, we want to have something there to be able to fight whatever microbes are able to enter our dermal layer. So the loose fibers are going to allow for phagocytes to kind of roam around just in case we have an abrasion that's going to allow for microbes to enter our dermal layer. So the dermal papillae that make up the dermal layer, they lie on top of what are called dermal ridges. And on top of these dermal ridges, we're going to get ridges in our epidermis. So they give rise to what are called epidermal ridges. We take these two ridges, the ones formed in our dermal layer, so our dermal ridges, and the ones that form as a result of the dermal ridges are epidermal ridges, and we collectively call these friction ridges. You probably know these more commonly as your fingerprint. So these are all the result of the dermal papillae in your dermal area, and then those that give rise in the epidermal layer. So friction ridges, they have more than one function other than your fingerprint pattern. These ridges allow us to grip something without it sliding out of our hand. And we know that we have the Meissner tactile corpuscles there, so they also contribute to our sense of touch. We have sweat pores, so when we actually leave a fingerprint, it's the result of our sweat. So we have sweat pores built in here along the way too. So friction ridges, basic thing, they're the result of our papillary layer and our dermal papillae. They give rise to our fingerprint, which is genetically unique to each one of us. You probably know that nobody except for identical twins has the same fingerprint. So it is genetically determined. They also contribute to our sense of touch and help us with enhancing the ability of us to grip something. The reticular layer is the second specific layer of your dermis. It's going to make up about 75 to 80% of the thickness. It's not going to be made up of loose connective tissue. Instead, it's going to be dense fibrous connective tissue. There's going to be a lot of elastic fibers there, so our skin does have some ability to stretch a little bit, as well as collagen fibers. And if you remember from your fibers, from the tissue chapter, these are your strongest fibers that we have. So it's going to help provide some strength and resiliency to our skin, as well as its ability to bind water to keep our skin hydrated. It also has a cutaneous plexus within it, and all that is just a network of blood vessels lying between your hypodermis and your reticular layer. You might also see some small areas of adipose tissue or adipose cells, but these are not predominant in this layer by any means. So the collagen fibers are the predominant fiber when we're talking about our dermal layer. And these collagen fibers run in the directions as shown here in red. So typically they run longitudinally if we're talking about skin in our limbs. If we're talking about around our neck or a trunk area of our body, collagen fibers typically run in a circular pattern. They're really important to surgeons, maybe not so much us, but if we make a cut that's parallel to these lines, it's going to help the skin gape less and it's going to heal quicker. So again, these cleavage lines are the result of the way our collagen fibers run within our skin, specifically our dermal layer. We have another thing that are called flexure lines. So if you've ever been to a palm reader, that's basically what a palm reader is reading, these different flexure lines right here. So these are dermal folds. They occur near our joints because our skin isn't super movable. Yes, we can pull it out and stretch it and it has some elasticity, but in areas where we have joints, where we have a ton of joints in our hand, we need our skin to have some flexibility. So we have these flexure lines and they're just dermal folds at or near the joints. So they help secure our dermis to deeper structures so that when our joints move and our skin stretches, we can move it easily. So they're visible in places where we have a lot of joints, our hands, wrists, fingers, our soles of our feet, and our toes. Now moving on to the pigments that contribute to our skin color, we have three: melanin, which we touched briefly on earlier, carotene, and hemoglobin as well. So melanin is the only pigment that's actually made in the skin itself. We talked about that's the function of melanocytes in our epidermis. And melanocytes, again, they just make and produce the melanin. They pump it into melanosomes, and the bulk of our melanin is actually found in keratinocytes. So their whole function is just to produce it and push it into the keratinocytes so that they can have UV protection. What is melanin made from? It's made from an amino acid called tyrosine. And we already talked about the melanosomes and its function and protection against damage from UV radiation. The more sun we're exposed to, the more melanin that'll be produced. It is a dark pigment, so that's why we appear tanner if we go out into the sun. Our body's producing more melanin to help protect us against the UV radiation. There are two different forms, so there's a reddish yellow form, and then it varies along a color spectrum until we hit brownish black. So the type and the color of melanin that our melanocytes produce is going to help determine our skin color as well. And if we're talking about a freckle or some sort of moles that appear darker, those are just going to be local accumulations of that dark pigment, which is melanin. The second pigment that can be found or contributes to our skin color is keratin, and that's going to be ranging from yellow to orange. Where is this pigment most obvious is going to be in the palms and the soles of our feet. It's going to accumulate in the outermost layer of our skin, so the most superficial layer, stratum corneum, as well as our hypodermis. It can be converted to vitamin A. If we are low on vitamin A, we would convert it to that. That's going to help with our vision as well as the health of our epidermis. And the final pigment, the final pigment here that you need to worry about that contributes to skin color is hemoglobin. So this is also the pigment when we talk about blood that's going to carry oxygen throughout our body. If hemoglobin is oxygenated, it is the scarlet red color. if our hemoglobin is not bound to oxygen, it's going to be a dark, deeper red color. So the pinkish hue of our skin is really going to be due to whether or not we're carrying heavily oxygenated blood. And if we have lower melanin levels, our hemoglobin or the reddish scarlet color is going to be more prevalent. So if you skin is more transparent, you're going to be able to see the hemoglobin pigment color uh, more clearly. You also need to be aware of alterations in skin color and how these can be indicative that you have some sort of disease. So go through your textbook and kind of take a look at cyanosis. That's when your skin has a blue hue to it. That's going to mean you have low oxygenation of your hemoglobin. Paler or blanching or pale color is going to be the result of a bunch of different things, not just disease. So, for example, if we get really upset, our blood might travel somewhere else, and it's going to cause us to appear pale, as well as fear. Different types of anemia can contribute to this as well, and low blood pressure. Erythemia, so this can be the result of fever, hypertension, or inflammation or allergy. So you'll see redness in certain areas of your skin, especially your face. And then jaundice, you'll get yellowing in the whites of your eyes, and that can be indicative or indicative of having a liver disorder. We are not going to touch on hair at all, like I told you. Um, so I put that here. Don't worry about this slide at all. The only thing you really need to know for this was the lab. You need to identify the root and the shaft. And that a hair is typically associated with an erector pili muscle, and you do not need to know anything about this. And I put in here, don't worry about 5.6 nails. So when we meet again in lecture, we'll talk about the different types of glands. And that's it.